After studying this module, you shall be able to know the differences between simple and conjugated proteins, categorize conjugated proteins on the basis of prosthetic group, learn the role of metalloproteins, know the coordination chemistry behind metalloproteins, identify the 4 classes of electron transferases, Flavidoxins, Cytochromes, iron sulfur proteins and blue copper proteins. Several types of proteins are available in nature, each of which is associated with different components and hence belong to various categories. Proteins containing only amino acid residues and no other chemical constituents are termed as simple proteins. Examples include enzymes ribonuclease A and chymotrypsinogen, whereas proteins that contain both chemical components and amino acids are called as conjugated proteins. The non-amino acid component of a conjugated protein is commonly called its prosthetic group. Further classification of conjugated protein is on the basis of chemical nature of their prosthetic groups. Therefore, proteins containing lipids are termed as lipoproteins, those containing sugar groups are termed as glycoproteins, and the proteins containing metal ions are called as metalloproteins. This has been demonstrated in figure. Number of proteins contain more than one prosthetic group. Usually the prosthetic group plays a significant role in the protein's biological function. Metal plus protein equal to metalloprotein. This is the figure of metalloproteins. Ever since the first X-ray crystal structure of a protein, sperm whale myoglobin, indicated the presence of an iron atom. Metalloproteins drew immense attention of biochemists and chemists. They account for nearly half of all proteins in nature, and the protein metal binding sites are responsible for catalyzing important biological processes such as photosynthesis, respiration, water oxidation, molecular oxygen reduction and nitrogen fixation. This is demonstrated by the active site structures of the enzymes involved, which are often reminiscent of minerals. Although metal centers form the basis of reactions, the protein matrix regulates reactivity. Function of metalloproteins. Metalloproteins are a class of biologically important macromolecules and account for nearly half of all proteins in biology. They are responsible for performing some of the most difficult yet important functions, including photosynthesis, respiration, water oxidation, oxygen transport, electron transfer, oxygenation and nitrogen fixation. These diverse functions of metalloproteins have been supposed to depend on the ligands from the amino acid, coordination structures and protein structures in the immediate vicinity of metal ions. Huge amount of efforts have been devoted towards understanding the structure and the function of metalloproteins. It is estimated that approximately half of all proteins contain a metal and about 1/4 to 1/3 of all the proteins are supposed to have metals in order to carry out their functions. It is due to the presence of the metal ions that allows metalloenzymes to perform functions which cannot be performed by the functional groups found in amino acids. Metalloproteins deal with almost all aspects related to intracellular and extracellular metal binding proteins, including their structure, properties and functions. The biological roles of metal cations and metal binding proteins are endless. Hence, metalloproteins play many different roles in cells such as storage and transport of proteins, enzymes and signal transduction proteins. Coordination chemistry behind metalloproteins. The metal ions in the metalloproteins are typically coordinated to nitrogen, oxygen or sulfur centers that belong to amino acid residues of the polypeptide chain incorporated in the protein. Amongst all, some important substituents are the imidazole, thiolate and carboxylate groups in the histidine, cysteine and aspirate residues respectively. Besides this, the peptide backbone also provides donor groups which include deprotonated amides and the amide carbonyl oxygen centers. Moreover, a large number of organic cofactors also act as donor to the metal ions, such as the tetradentate N4 macrocyclic ligands incorporated inside the heme protein and inorganic ligands such as sulfide and oxide. 4 classes of electron transferases. In most of the biological processes such as photosynthesis, respiration, collagen synthesis, steroid metabolism, the immune response, drug activation, neurotransmitter metabolism and nitrogen fixation, electron transfer reaction proteins play major roles, out of which both respiration and photosynthesis are extremely significant processes as they provide most of the energy that is required to sustain life. Metalloproteins, which has a single type of redox factor, can be classified into two general groups: electron carriers and proteins involved in the transport or activation of small molecules. It is assumed that proteins that function as electron transferases typically place their prosthetic group in a hydrophobic environment where, in addition to ligand, hydrogen bonds may be provided with the help to assist in stabilization of both the oxidized and the reduced form of the cofactor. In order to minimize the inner sphere reorganization, metal ligand bonds remain intact upon electron transfer. Many of the complex metalloenzymes containing multi-site such as cytochrome C oxidase, xanthine oxidase and the nitrogenase iron molybdenum protein have redox centers that function as intramolecular electron transferases. There are four classes of electron transferases: blue copper proteins, cytochromes, flavidoxins and iron sulfur proteins, each of which contains many members that exhibit important structural differences. Flavidoxins. The flavidoxins are proteins with molecular weights in the range of 18 to 13 kilodalton and are found in many species of bacteria and algae. These proteins contain an organic redox cofactor, flavin mononucleotide, which is also known as FMN cofactor, that is found at one end of the protein near the molecular surface, but only the dimethylbenzene portion of FMN is significantly exposed to the solvent. FMN can act as either one or a two electron redox center. In solution, the semiquinone form of free FMN is unstable and disproportionate to the oxidized form called quinine and reduced form called hydroquinine. This has been represented in figure. Hence, free FMN works as two electron agent. On the other hand, FMN in flavidoxins can function as single electron carrier. This is easily differentiated on the comparison of reduction potentials for free and protein bound FMN. Evidently, the protein medium is considered to be responsible for this drastic alteration in oxidation state stability. It was also concluded from the NMR study of L-decenni flavidoxin, quinone, semiquinone and semiquinone hydroquinone electron self-exchange rates that the latter is approximately 300 times faster than the former, in keeping with the view that the physiologically relevant redox couple is semiquinone hydroquinone. Cytochromes. Out of the four classes of electron transferases, the cytochromes are the most thoroughly characterized class and were among the first to be identified in cellular extracts due to their unique optical properties and prominent intense absorption in the region of 410 to 430 nanometer, known as the Saurit band. By definition, cytochromes are proteins containing one or more heme cofactors. These proteins are generally classified on the basis of heme type. There are three most commonly encountered types of heme. Heme A possesses a long phytyl tail and is found in cytochrome C oxidase. Heme B is found in B-type cytochromes and globins. Heme C is covalently bound to C-type cytochromes via two thioether linkages. Figure represents cytochrome C. The real challenge lies in the nomenclature of these cytochromes. Some of them are designated according to the historical order of their discovery. Example, cytochrome C2 in bacterial photosynthesis. Others are designated according to the A max of the band in the absorption spectrum of the reduced protein. Example, cytochromes C55. Of all, cytochrome C is the most dominant in nature. Ambler divided these electron carriers into three classes on structural grounds. Class 1, cytochrome C contains axial His and Met ligands with the heme located near the N-terminus of the protein. These proteins are globular, and X-ray studies of class 1 cytochrome C from a variety of eukaryotes and prokaryotes clearly demonstrated an evolutionary preserved cytochrome fold with the edge of the heme solvent exposed. Cytochromes possess positive reduction potential 200 to 320 millivolt. Class 2 cytochromes C are found in photosynthetic bacteria where they serve an unknown function. Unlike class 1, these C-type cytochromes are high spin where the iron is 5 coordinate with an axial histidine ligand. The vacant axial coordination site is buried in the interior of the protein. These proteins, generally referred to as cytochrome C, are the 4x helix bundles. Class 3 cytochrome C, also known as cytochromes C3, contains 4 hemes, each ligated by 2 axial histidines. These proteins are found in the restricted class of sulfate reducing bacteria and may be associated with the cytoplasmic membrane. The lower molecular weight of cytochrome C3, approximately 14.7 kilodaltons, require that the 4 hemes be much more exposed to the solvent than the hemes of other cytochromes, which may be in part responsible for their unusually negative -200 to -300 millivolt reduction potentials. These proteins possess many aromatic residues and short heme-heme distances, two properties that could be responsible for their anomalously large solid state electrical conductivity. Iron sulfur proteins. The iron sulfur proteins play essential role as electron carriers in nearly all living organisms and take part in plant photosynthesis, nitrogen fixation, steroid metabolism and oxidative phosphorylation, as well as many other processes. The simplest of all iron sulfur proteins are the rubridoxins, which are primarily found in anaerobic bacteria. Rubridoxins are small proteins, approximately 6 kilodaltons, and contain iron ligated to 4-cysteine sulfur in a distorted tetrahedral arrangement. The reduction potential of iron sulfur proteins are typically quite negative, indicating a stabilization of the oxidized form of the redox couple as a result of negatively charged sulfur ligands. Flavidoxins, 2 iron 2 sulfur, are the other class of iron sulfur proteins, approximately 10 to 20 kilodaltons, which are found in plant chloroplasts and mammalian tissue. The structure of Spirulina platensis Flavidoxins confirmed earlier suggestions based on EPR and Moss pair studies that the iron atoms are present in a spin-coupled 2 iron 2 sulfur cluster structure. The optical spectra of all iron sulfur proteins are very broad due to numerous overlapping charge transfer transitions that impart red, brown, black colors to these proteins. In contrast, the EPR spectra of iron sulfur clusters are quite distinctive and have significant value in the redox chemistry studies of these proteins. 4 iron clusters, 4 iron 4 sulfur, are also termed as Flavidoxins, which is found in many strains of bacteria, and in most of these bacterial iron sulfur proteins, 2 such clusters are present in the protein. These proteins are small with molecular weight of 6 to 10 kilodaltons and have reduction potentials in the 400 millivolt range. Each of these clusters contains 4 iron centers and 4 sulfides at alternate corners of a distorted cube where each iron is coordinated to 3 sulfides and 1 cysteine thiolate. The irons are strongly exchange coupled, and the 4 iron 4 sulfur cluster in bacterial Flavidoxins is paramagnetic when reduced by 1 electron. The so-called high potential iron sulfur proteins HIPIP are found in photosynthetic bacteria, exhibit anomalously high, approximately 350 millivolt reduction potentials. The C-vinosum HIPIP, which is approximately 10 kilodalton, the structure demonstrates that HIPIPs are distinct from the 4 iron 4 sulfur Flavidoxins, and the reduced HIPIP structure is significantly distorted, as is also observed for the structure of oxidized P-aerogens Flavidoxins. In addition, the oxidized HIPIP is paramagnetic, whereas the reduced protein is EPR silent. Blue copper proteins. The blue copper proteins are characterized by intense S-cysteine copper charge transfer absorption near 600 nanometer, an axial EPR spectrum displaying an unusually small hyperfine coupling constant and a relatively high reduction potential. With few exceptions, example photosynthetic organisms, their precise roles in bacterial and plant physiology remains obscure. X-ray structures of several blue copper proteins indicate that the geometry of copper site is approximately trigonal planar. In all these proteins, 3 ligands, 1 cysteine, 2 histidine, bind tightly to the copper in the trigonal arrangement. Differences in interactions between the copper center and the axially disposed ligands may significantly contribute to variations in reduction potential that are observed for the blue copper iron transferases. For example, E0 equal to 276 millivolt for A azurin, whereas that for P vulgaris plastocyanin is 360 millivolt. These differences in bond lengths are expected to stabilize copper in azurin to a greater extent than in plastocyanin and result in lower E0 value for azurin. Summary: simple proteins containing only amino acid residues and no other chemical constituents. Conjugated proteins contain both chemical components and amino acid residues. The non-amino acid component of conjugated protein is commonly called its prosthetic group, and the further classification of the conjugated proteins is on the basis of the chemical nature of their prosthetic groups. Proteins containing lipids are termed as lipoproteins, those containing sugar groups are termed as glycoproteins, and the proteins containing metal ions are called as metalloproteins. Metalloproteins account for nearly half of all the proteins in nature, and the protein metal binding sites are responsible for catalyzing important biological processes such as photosynthesis, respiration, water oxidation, molecular oxygen reduction and nitrogen fixation. Metalloproteins deal with almost all aspects related to the intracellular and extracellular metal binding proteins, including their structure, properties and functions. Metalloproteins play many different roles in cells such as storage and transport of proteins, enzymes and signal transduction proteins. The metal ions in metalloproteins are typically coordinated to nitrogen, oxygen or sulfur centers that belong to amino acid residues of the polypeptide chain incorporated into the protein. There are 4 classes of electron transferases: blue copper proteins, cytochromes, flavidoxins and iron sulfur proteins, each of which contains many members that exhibit important structural differences.